it's a paradigm. Oh, good. It's a paradigm that incorporates the complex nature of evolution in the biological system. And I'm going to show this in a little bit, that the other two elements of the Stockholm paradigm, which is the higher elements of this paradigm, oscillation and taxon pulses, are really derived or a consequence, or they are really emerging properties of the ability of organisms on this planet to do ecological fitting. So, and we have shown this through modeling, and it's becoming more and more evident that this is the real scenario right now. So I'll be talking a lot about ecological fitting, a little bit about oscillation, and not much about taxon pulse, although at the end we'll talk about that a little bit. But keep in mind that ecological fitting is the basis in my perspective of the whole paradigm. This is what makes things work on this planet. So I have a whole bunch of slides. I've tried to go relatively fast through most of them, but I'm going to assume that you guys have heard a lot about the Stockholm paradigm from other speakers before me, so I don't have to explain or go through all these elements in detail, but I'll talk about them a little bit. Sorry. So ecological fitting is just that, what's written in there. It's what the species can do with whatever information it has. It has to handle challenges and opportunities in order to survive and diversify on this planet using whatever it has at that specific moment. There's no magic mutation as some of us used to think. So we species have to deal with the problems with whatever they have, and we know this is real, right? This is a suggestion based on the accumulated knowledge we have on the modeling and so on, that life was probably associated with the origin, that ecological fitting was probably associated with the origin of life, because when we start reading about flexibility of organisms, we find that molecules, especially organic molecules, are highly flexible as well. There is a saying that there's a terminology for enzymes that indicates that, and the terminology says that enzymes are usually promiscuous. More than 95% of the enzyme are promiscuous. That means that they can catalyze in both ways, and they can catalyze many different chemicals, and that makes the enzymes more flexible. And so we think that the origin of life was only possible because of flexibility of the systems that compose that thing that we call the origin of life. The metabolic pathways, the membrane, the information content, everything was put together because they could come up together because they had some flexibility to act together, right? And that continues through the complex system that we call life on this planet. So ecological fitting will be the most fundamental property of life. And then we have multicellular animals appearing because of this ecological fitting. We have associations with it, which is interactions, which are absolutely fundamental for life on this planet, as a consequence of the ability of the elements to do ecological fittings and so on. If you add time and evolution, you end up having oscillation. Oscillation is an emerging property of individuals or species that can actually do ecological fitting, and I'll show this a little bit a little ahead. And if you add space, then you have taxon pulse, especially space like on this planet, which is a highly variable space, and it's a cyclic. We have cyclic disturbance. We have cyclic establishment of barriers and elimination of barriers and so on that would produce naturally taxon pose, and it's also considered a emerging property in the complex system of biology and evolution. So this is the basic scenario. So I want to talk, I will be talking a lot today about this two components that have to be considered in evolution in order to actually understand evolution. And there is a lot of misconception, especially based on capacity and opportunity. So evolution is about the accumulation of evolutionary novelties that are inheritable from generation to generation, and that will allow species to handle expansion and diversification. I went back. So evolutionary novelties are accumulated through anagenesis, and inheritance will tell us about the history of the species. So a lot of different anagenetic changes that happen in the ancestors are accumulated in the species today and in the past. And cladogenesis are just a matter of opportunity in expansion and diversification. And these are the two elements that I think we have to talk about, and I will be talking about a lot, which is capacity of the individual and opportunity. This is how I envision the capacity space, which is just a portion of what I call the information space. I know we've been going back and forth about this in our discussions on the inheritance of information, but if we accept that the whole thing is called information space, and information space would be anything that is inheritable in a species that can go through generation, that includes genetics, that includes education, that includes change in the network of genes, the pleiotropy, epigenetics, and so on. Inside of this information space, there's a special article, the fundamental capacity space, or the capacity space as a whole. The capacity space is all that space that a species can actually use to solve problems. Within that capacity space, there's a smaller area that I call realized capacity space, which is what the species is actually using. So the species is actually using whatever is necessary to survive under a specific system or selective region, a specific selective force. The drawings I have down here are trying to explain that a little bit. So in red, in the first giraffe that we have there, in red, we have the information, the whole thing is the information space. We have in yellow the area that is not being used. It's the part of the information space that is not in use. And whatever it's in red, it's part of the capacity space that giraffe is actually using to survive. The capacity space which is a property of the organism, when contrasted with the nature of the conditions and the selective force, will generate, the capacity space will define the fitness space. The fitness space is what the species can do based on its capacity space, but it's not necessarily doing it right now. In red, on the second figure here, is whatever it's being done. This is called the realized capacity space. So that's what the species is doing or using from its information space, from its original capacity space, or the whole capacity space to survive in a certain condition. But if the condition changes, that does not mean that the species will disappear if it doesn't evolve a new feature, a new trait. It can simply use different areas of the fitness space in order to survive under different conditions. So that's what I'm trying to show here when I show the real small red dot moving away from the original big dot, showing that giraffe, even though there are no trees, they can still eat grass because they're capable of changing the behavior, which is part of the original capacity space, although it was not using at that time. So this is how I envision it. In this, the capacity space evolves, and we know this from genetics, right? But again, it's not only genetics. With time, you have accumulation of novelties that are incorporated into the capacity space that increase the fitness space of the species. And of course, if you don't use part of the capacity space, there's a chance that part of that capacity space will suffer erosion and we'll lose some. And that will be important to understand when I start talking about relationship in time with host and parasite. But the erosion will imply in the loss of the ability to handle changes in the environment or to colonize new areas or survive new hosts. Because if you need that area that was eroded or lost, it could be, and it's usually lost just by chance, not because there's a selective force forcing it to disappear necessarily, but that would have an implication on what the species can actually do. And we're talking about parasite, it can have an implication on what kind of host it can use. So let's keep that in mind and move forward. All right. So, and the second element that I showed you, capacity was the first one. The second one is opportunity or challenge. Let's use opportunity as the word to be used. If you don't have the opportunity to jump boat, if the boat is too far away, you're not going to be able to jump to another boat. So if you don't have the opportunity to use another resource to explore another host, or use a different environment to colonize a new environment, it's not going to happen. So the species are really sometimes capable or have the capacity to do much more than actually they are doing. And they will only show you that capacity if you provide the opportunity. So that's one of the big discussions we have in the Stockholm paradigm is some of the modelings that we do for distribution are based on whatever the species is doing right now. And species usually can do a lot more than that. I can after we're done discussing here, I can talk about the study we did with the Brazilian red pepper that have more ability or more capacity to live in different environments than actually it shows us. Yeah? It's originally from Brazil, from a tropical area, but it can live in temperate areas because the ancestors could do it. And we actually see that because it's an invasive species. I guess I already explained it. So that's what I'm going to be talking about today. So within this scope of opportunity and capacity, we're going to be talking about exploring, colonization, exploitation, and a little bit of differentiation, range expansion, which is the result of all this, and diversification. And you see that I have an arrow going back to exploring because this has to do with what we're talking about, replenishing the risk space. And the risk space is the number of potential pathogens that a certain species or all the species on the planet are exposed to. So since it's cyclic, there's a replenishing going on, and I'll show you in a little bit. So we are surrounded by parasites and pathogens all over. I mean, we can exemplify this by showing a plant and showing that there are a lot of insects trying to consume that plant also. That's one of the things that I learned when I started working on the Stockholm paradigm, is that all these different fields of biology are just the same. You know, we use different names, we use different terminology. We call it a special field. So parasitology is special, phyto insects that are phytophagous are something else, then invasive species is something else, but it's basically the same process. Basically the same things are happening, the same pattern are showing up, and we use different names. And you probably recall Vernon Thatcher. Vernon Thatcher said that if you really want to be a specialist on something, create your own field of science and create your own terminology. You're going to be the only one to know about it. So I think we're entering the time that we're going to be uniform, creating uniform terminology for all these processes, and not only terminology that are specific to a certain field in biology. So and this comes from conversations that I have with Dan and Eric. Every single species of host, and a parasite can be a host too, right? It's being exposed to pathogen all the time, and we have this thing that we call pathogen and parasites pollution, which is really part of the exploring part of this whole process. And I'm using a non-Commericidium as an example for obvious reasons. So this non-Commericidium managed to jump and survive on a fish. That at the immediate survival, it's exploring a new host. If it can actually survive that host, then it will start the colonization process. But sometimes, and most of the time, it doesn't, right? Sometimes there are specific clues to find the right host, but inevitably these guys are going to end up in the wrong host, or they're going to end up dying because they couldn't find any host. So this would be an unsuccessful exploration, an unsuccessful colonization. But if it survives, and I have a drawing of the a schematic drawing of a fitness space of a certain parasite, which is the guy with the flag in the hand there, the blue, and then the dark blue would be the necessary space in the fitness space of the parasite that is required for the parasite to survive on that host. So if there is a minimal chance for it to colonize, it may colonize. It will depend on a series of factors, but most of all on capacity. And we'll talk about this in a little bit. So colonization follows. This is the result of the first paper, the first model that Dr. Sabrina did with a lot of other people. But what we did basically was to simulate attempts of the parasite, which is indicated by the black points in this figure. What happened with this parasite when it was exposed, parasite population, there are many points in there. When they are exposed every generation to different hosts at different distances. And you can see this as a morphological distance, morphophysiological distance, or phylogenetic distance. So the green are the different hosts being presented at each time interval, and these are the time interval on the x-axis here. If the colonization occurs, then you have a red line indicating that the population of the parasite is living on that specific host. If you see down here, there's a green square, and then a red line indicating that the population in black, all these individuals here are living within that same host for a long period of time. So you have to do cross sections to understand that this is the present population of parasites. And this describes the phenotype of the parasite, which is pretty much analogous to what we call the capacity space. So all these hosts are being offered to this parasite living on this host, but they're not host switching. There was no successful host switching here. There was no successful jumping onto another host and expanding host range. On this model, we didn't follow the original host. So we didn't look for host range. We just looked for what happened to the parasite when it colonizes the new host species. And then we forgot about the old host and we followed the new one. And there are some interesting things that come out of that. One of them is that you do have many times not the parasite that is close to the host being capable of colonizing that host, but sometimes distant parasites are capable of colonizing the host. So there's a lot of chance going on in here, which individual with which nature of the organism will jump onto the host and try to survive on it. Sometimes, even if you're very close to it, because of the model, because of the simulation, the closest parasite, let's start using the term compatibility. The most compatible parasite not necessarily jump onto the new host, but a distant one. So, and usually you have a reduction in variability. There's a clear reduction on the makeup of the parasite population, but it will in time become more and more adapted or specialized or differentiated according to the selective pressure imposed by the new host. So let's move. So this is the end result, and it shows that this graphic on the right is especially interesting. This is the distance between the parasite and the necessarily fitness space to live on that specific host. And what we can see is that even though we have sometimes information, the capacity space of the new population being 0, I'm sorry, is this one? I got lost here. The capacity space, yes, being 0, we still have a good probability, a good chance of that parasite surviving on the new host. So on the model, the diversity of the parasite propagules colonizing the host can be 0. That means there is no variability. All clonal individuals, they're still capable of colonizing the new host. And so variability is not necessary to exist to allow colonization. The other thing that is interesting on this graphic is this blue area, which is the inability to colonize new hosts. So every host that is distance above a certain value, the parasite is not capable of colonizing that host anymore. So this makes sense when we see that there's something that people call host specificity. You're not going to be able to take a parasite of insects, maybe, and a nematode of insects, and they could survive in a very distant, phylogenetically distant group. So there are some gaps in there. So ecological fitting allows for a more efficient colonization of a wide range of hosts, but not everyone, not every host. So this is important to remember. So to understand capacity, we have to understand compatibility. And on the right, there is a matrix of parasites on the left and indicated by different colors on the right. So these are different orders of fishes, parasite monogenes from South America. And all these spots are parasites that are found on these specific groups. And there's no name down there because it wouldn't make any sense. It's too difficult to read, but it shows that there's clearly some difference in compatibility. If it was compatibility would be maximized, I'm sorry. If compatibility would be maximized, if there was no blue area on that graphic that we show as a result of the first model, we should have dots disorganized on this plot. You know, species of parasites from siluriforms living on characiforms and perciforms and so on. But this doesn't happen. That means that there is some level of compatibility that is clearly different among different lineages of hosts and parasites. And this would be fantastic, well, absolutely important to know how to estimate compatibility if we want to deal with, for instance, emerging infectious diseases. If we can predict or estimate the compatibility between host and parasite will be able to apply what we have published this in 2014 as a consequence of a meeting we had in Ogallala. We could apply the access portion of DAMA. And the most fundamental, and this has been known since the beginning of the proposal of DAMA, which is, again, I am assuming that you know a little bit what that is phylogenetic triage. And phylogenetic triage is a way of incorporating all traits that are related to the compatibility between hosts and parasites and synthesizing it into the phylogenetic distance of hosts and parasites, right? Parasite groups and host groups and how they match. And that's a very smart way of doing that because of evaluating compatibility, because we know that groups that become associated are members of different clades in the life of this planet. That means that they have different qualities, different characteristics, different nature of organisms. And we would not be able to identify specific traits associated to compatibility, right? So viruses and mammals is one thing, and nematodes and fishes are a completely different thing, right? Different elements, different traits. There's no way we could estimate compatibility among hosts and parasites using a specific set of traits. But that's when I go in. I have to mention this. I think Dan would like that, because that's something that he's been saying for a long time. We like to have simple things, and things are usually not simple. So chaos is the law of nature. And when I start thinking about this phylogeny thing, and I thought that even though we do have phylogeny as a unique central element to define compatibility, we have to remember that again, the rate of change of individual clades is completely different. So the rate of change of a complex organism, like a multicellular organism, is completely different from the rate of change of its nature of a prokaryote or a virus. So it's not a simple task to do this phylogenetic triage. And I've been reading about this crazy thing about chaos, which is the chaos theory, and it makes a lot of sense to me when I start reading it. For physics, there are many elements that compose the theory of chaos and are used to identify chaotic systems. And there are many different chaotic systems that are being recognized these days. But one of the things that I called my attention was that small differences in initial conditions may generate completely different outcomes. So this is what this animation is showing, is very small differences in the way that the pendulum starts the dynamics that you're seeing here. We'll end up with completely different pathways, different routes that they follow. And this is in physics, right? And one way of identifying the difference between the vectors of each one of these situations is by applying what they call Lyapunov exponent. Don't ask me to explain it, but it's a way of showing how predictable a system is, how predictable a movement is, how predictable a process is. And in physics, very small differences will promote completely different things in a very short period of time. If we look, and this was initially, this is the butterfly effect that you probably heard about or saw in movies and so on, although in movies it's kind of funny, but that confers a predictive power to that indicates what is the limit of the predictive power of a deterministic system, which is the chaotic system that becomes randomic with time. So what the chaos theory says is that you have for some systems you have a level of deterministic period that can be used to do some predictions about the system, but that the ability to predict, even though it's a deterministic system, is lost after some time. But for instance, in chaotic electrical circuits, it's like 1 millisecond. After that, you can't predict anymore. I mean, we know about weather systems, right? So I can predict up to a few days, 15 days, but after that, it's becoming more and more unpredictable. For the inner solar system, it's 4 to 5 million years. What about for host-parasite compatibility? What I expect, and that comes one of the books that I like the most, then, is the fact that the biological system, with time, will not express all the changes that happen to it. So it's like saying, even though if you have a lot of mutation going on in one system, in one lineage, not all these mutations will remain. That means that there's some level of limitation or constraints for changes in the biological systems. And I kept thinking about this, and I said, well, that's a little bit different from what we see in a physical system, but there are no really a lot of constraints in a physical system. I may be wrong. I mean, I have Sabrina to correct me if it's necessary. So if we look at evolution it's in fact strongly conserved, and that makes sense, because if we have ecological fitting as a process happening in evolution that depends on whatever the species had and was accumulated by its ancestor in the past, that means that I'm not gaining a lot of new stuff, no, not of new traits. I have to use old traits to deal with the problems I have. So if you look at a morpho-space analysis using phylogeny, the red, for instance, is not spread all over the morpho-space area. It's limited to a certain area. That means that there's a lot of conservatism going on in evolution, which is good for us. In some of our models, we showed that the capacity, if you have a virus that is very fast, the capacity to go back to jump to a new host and then go back to the previous host is retained after colonization of the new host. And this is what I'm showing on these two figures B and C here. On figure A is, this is a slower rate of evolution than the two right panels. So if you have a relatively fast rate of evolution, which is something we see in viruses, you leave a tail. One indicates the phenotype necessary to survive on the original host, and 0 is the phenotype necessary to live on this new host. And you can see that the colors are showing different distribution of phenotypes within the virus population that goes from 1 to 0, I'm sorry. But even though it reaches 0 in this dark brown, it still have a tail down to the original host. That means that some part of this viral community or population can recolonize the host. So we are retaining capacity space, even if it's a virus, and we have seen this in COVID, and I'll show you in a little bit again. So let's speak about that capacity of predicting the evolution of the whole space system that I showed before, which is 5 million years. If we look at COVID, how COVID behaved, these are some of the, I'm trying to get some space to see what I'm showing here. But where COVID can survive on this phylogeny, the distance between the most basal host and the most distal host on this phylogeny that is being used by COVID is approximately 150 million years ago, 50 million years. So that means that the elements and the traits that are associated to the compatibility between host and parasite, host and pathogen, are conserved to the point where there's still compatibility between hosts and parasites after 150 million years. And let me show you a study that I did some years ago using this group of fishes. These are freshwater groupers, freshwater cyanids of the subfamily Pachyurinae. And these are the Pachyurinae here, and these are the Plagiausium, which was considered at that time as being a different subfamily, but we now know they are all the same thing. It's just that Plagiausium are an early branch of the evolution of Pachyurinae, which is a group of croakers that live in freshwater South America. And the study we did back then suggested that this group, which is a member of Cyanidae, and they are marine, typically marine animals, they colonized freshwater South America approximately 30 million years ago. And they start spreading and diversifying into the different hydrographical basins we have. But the interesting thing that happened here happens also with invasive species as they spread or as they expand in geographic distribution, they lost parasites. So they in the Amazon, that's where they colonized, the first area they colonized. They have like 11 different species of Diplectenidae and Herialeotherma, which is a different genus of monogenes. As they spread, they start losing parasite. When they moved from the Amazon into the San Francisco River and into the coastal Atlantic basins in Brazil, they lost completely the original parasites they had. And the same thing happened when they moved into the Paraná River. Paraná is in blue, is in yellow, and in green is the Uruguay River, which is in fact part of the Paraná, but they also lost other parasites. Interesting enough, they spent between 30 million years until today, and they still don't have a single species of monogene from freshwater or from primary freshwater fishes. So there's this big barrier of compatibility, like I showed or suggested before. But after around 13 million years of spread, they colonized the coastal area of the Atlantic coast of Brazil, which is composed mostly of very small and short rivers. In my understanding, that increased the opportunity of contact in the estuarine area between marine cyanids, which is this one that is evolving in freshwater Brazil, with marine cyanids, with freshwater cyanids and marine cyanids. And what happened in here is that one species that parasitize, or one lineage that parasitize marine cyanids, are capable of jumping into this lineage of freshwater cyanid that have been in freshwater for a long time. So the distance between this host, which is a Mantoceros, apparently a group, most likely the group that donated the parasite to the freshwater species, the difference between these two lineages of fishes is 43 million years. In the Paraná system, they also lost all the parasites, but funny enough, this is a Plagiausium, which is that basal lineage of freshwater cyanid that evolved and dispersed independently from Pachyurinae, which is the one that we are focusing on. But this guy donated one species of Diplectenum that is also found in the Amazon, that is also found in the Paraná River, but that is not found in the Uruguay River. And in fact, in the Uruguay River, there is no species of Plagiaulus. There's no species of this group of fish. So they are present in the yellow, but they're not present in the blue area, in the green area. So again, they split about 20-something million years ago. They evolve and they disperse independently, but once they had the opportunity, probably in the Paraná system, there was an expansion of this parasite into the Pachyurinae, which dispersed independently into the Uruguay River, but Plagiaulus did not. So the parasite is really telling us a little bit about the biogeography of the fish. Again, compatibility was retained between 8 to 30 million years. So that tells us about the predictability of using phylogeny to do the triage, a phylogenetic triage of host and parasite, to identify potential parasites that can be a problem to whatever host group you're working on, which is kind of interesting. I think it's a very interesting area to work on. I'm going to try to go faster. So this is a study done by Elvira De Bastiani. She's in Los Angeles right now. And she tried to simulate the idea that capacity is retained through time, or compatibility is retained through time in the evolution of the groups. But there's a little bit of erosion going on, and the erosion is inevitable. We lost capacity and we lost some hosts, so we lost part of the parasites. So we lost part of the parasites. They're not accommodated to that lineage of hosts that died. And I'm not saying that it's a different species or not. It's just that group was submitted or subjected to a different selective force. And what she suggested, and that's one of Sabrina's student, Sabrina is coordinator of this project, is that the compatibility between host and parasite was lost through time, right? So they incorporated this into an IBM, an individual-based model. They analyzed many simulations analyzing host expansion, host specialization, what they call, putting it all together, as host switching. So you can expand your host range, you can speciate or not. No? You can expand or speciate or just expand. And when they compare these values with real values, network values, parameters, including richness of parasites of existing real systems, they found out that it varies from a little bit above 0% up to 15% of the percentage of individuals that explore the new host and survived. So host switch, exploration happens all the time. Successful host switching that we can see today go up to 15% based on these examples. But that shows also how different the systems are and why we need a more generalized proxy for compatibility. And I think we're starting to see that we already knew that phylogeny was a good proxy, but it's probably better than we thought before, because if we have 150 million years of difference between hosts, not enough to break compatibility 100%, then it must be a good parameter to analyze or to estimate compatibility. So once colonization happens, what we did in this study was to, again, Sabrina was our physicist in charge, and Sophia is one of our ex-students. The study, she concluded that propagule size or the number of Vibrio in terms of viruses or number of larvae that arrives into another host will make a difference in the success of colonization of a new resource or a new host. And this is what it's showing here. So reproductive rate was important, novelty rate was important, but propagule size was especially important. So this would be like mutation rate, novelties rate would be like mutation rates for viruses. But if you add all these three together, and this is the gray line in each one of these graphics, this is the synergetic effect of these three maximized. The maximized values of these three factors will almost increase dramatically the ability of colonizing new hosts. And if we think of viruses, this is what it is, right? This is what viruses are. So I'll jump this. This is just talking about the oscillation in the size of the capacity space, not because the individual loses information, but that there's a loss in the population composition of diversity, and diversity is part of the capacity space. So within the capacity space, you have the individual capacity space, and then the combined capacity space of a population of parasite will define the capacity space of the population. If you usually have a reduction, and this is known for viruses really well, and I think it's probably widespread in any ecological change we see in the planet, not 100%, but a lot of them are associated to reduction in variability, reduction in the size of the capacity space. But with time, like the graphic is showing on the left, capacity space increases with time, with accumulations of novelties, with accumulation of mutations for viruses, if you want to think like this. I'll jump that. This will define the way that species are exploring the resources, right? So even though we have a reduction in variability in the colonization of the new host, you have a recovery of the capacity space, which means that you probably have some differences in the fitness space of that species to handle host switch, to handle expansion to other hosts. And Marina, which was one of my master's students, she did a Ph.D. later on in Sweden. She worked with Sabrina as well. She did this study where she now retained the original population and trying to understand what happened with the expansion of the parasite into different host species and watching what happened to the population of parasites within these different host species. And I have an animation that shows pretty much what happens. So that's one parasite, one host in each color, right? And the color indicate the same parasite. The branch indicate different hosts. That's what it is. Because down here on the x-axis, what you have is a different nature of the host. And what happened? She exposed one parasite to 10 different hosts within a petri dish, if you want to call it. And she saw that the parasites were able of colonizing or using that host and then expanding to three hosts, and then going into one color line here, reducing the number of hosts being used and specialized to one host and so on. This variation in the specialization of the parasite, one host, three hosts, two hosts, one host, three hosts, so on, is called oscillation. So by allowing the parasite to use ecological fitting to colonize different hosts and exposing the hosts together, you naturally generate oscillation. You don't need space to produce oscillation. Oscillation appears to be the consequence of this fluctuation of the capacity space of the parasite population. So that's pretty cool, but that's also how variants are likely formed when we talk about parasites, right, or pathogens. And many times you see people talking about, special medical people talking about, oh, there has been a mutation that allowed the parasite to colonize the new host, but they forget And this would be on the left side, the original parasite, the original virus, the distribution of diversity of the original of the virus on the original host. On the right is the new host. And what we see here is that most of the times, every time you have a host switch or expansion to a new host, you have some features, some traits, or some abilities, some elements of the population of the original virus that becomes more important for the survival on the new host. To the point that if I go back to this original host, let's say this monkey, in trying to describe the diversity in this monkey of that specific virus, I will not detect that specific variant or that specific variant of the viruses in the original host, but it will show up very fast in the new host. For an MD or for a person that doesn't understand what's going on, that variant, the colonized new host, will look like a new mutation. But the chances are that that specific variant was already present in the old host, and it just become more prevalent in the new host. And this is what probably happens to COVID, right? What we're doing here is we provided opportunity for a viruses that has the compatibility of 150 million years with different species of mammals. And we ended up having things like this, colonizing different species of domestic zoological animals and wild animals and coming back to us. And the most impressive one is what happened to Omicron, right? This very long branch indicates a change in the selective region of the virus. And we don't know which host is that, but it came back to us. And it went back to wolf again. And it probably will come back to us, and we'll go back to wolves again. And that will happen many times. And it's probably going to happen on every single group that you see here, as long as there are opportunities associated to it. This is just a bar plot graphic showing Omicron, the branch of Omicron up here. This is the original branch of Omicron around here. That's why we suggested that Omicron indeed was using a different host and not humans, like has been suggested in the past. And this is what you see when you look at the plot of the mutation rate, accumulated mutation rate for Omicron is much higher than that observed for the other variants, probably because it was using a different host until it reached the humans again and start accommodated to humans again. I don't want to talk about this. So just to finish, and I think I talked too much already, just think about what happens when we combine different possible host species, different selective region for each one of these hosts, different geographic regions, different connective rates between these regions. We are really having a fantastic machine to produce new variants. We probably have a lot more variants than we actually recognize these days, and we will have more. That means that we're going to be replenishing what we call the risk space for a long time. Just to finish, these are the last slides, I promise. This is the way I envision the evolution of host and parasites with this big pathogen pollution or parasite pollution, giving two things, opportunity and compatibility. So I'm trying to show opportunity as this Brownian movement, suggesting propagules, exploring the different areas of the tree of the host that I'm showing here. There's some extinction of hosts here. There will be some vicariant going on. But the compatibility model does not necessarily follow the opportunity model. So remember, opportunity is the ability to contact the new host, to contact a new resource, to explore new areas, and compatibility model is the ability to do that. And they're not synchronized. They're completely off sync. So at the beginning, that first host was being exposed to the specific cloud of propagules. And at this moment, a vicariant barrier appears and originated different lineages of hosts. And then we have two modules of opportunities showing up, appearing, because one module does not have access to the other module. So parasites from the right cannot access hosts from the left and vice versa. And they evolve this way. Compatibility tends to change due to evolution, due to erosion. The opportunity model has split, but compatibility still exists, although there are some changes in it. Does not necessarily means that the parasite from the right is not able to colonize in the yellow, for instance. And another vicariant event may happen. They'll create a new opportunity module. The compatibility module continues to be unique, although there will be a single one, although there may be some changes. And with time, there will be changes and there will be splits in the compatibility model as well. But this is where the trouble is. This is where taxon pole centers. If we eliminate the barrier down there, we expand the opportunity module. And now we have retro-colonization or symmetrical colonization, generating new lineages and new variants of parasites or pathogens and so on that are going to recolonize lineages that it has evolved with, or where it has survived for a long time. Someone is sending me messages, so I'm just checking if it's not one of you. Anyway, this is how we call replenishing the risk space. So there will be replenishing due to geographic, due to hosts, different hosts being exposed, different lineages being formed and coming back to us or to whatever species we're interested on. I'm sorry if it took me this long to tell you everything. I'm sorry if I didn't make myself clear. I hope I can make it a little bit more clear if we talk a little bit from now on. But this is just a list of conclusions that I come up from this presentation. Like I said, there's so many different things to talk. Ben knows that I would be here for 4 hours talking, but I don't have that time. I almost did that in Hungary, Dan. So thanks, guys, for your attention, and I hope it was clear enough so we can talk.
Thanks a lot. Yeah, it was really good. And any questions from anybody out there in internet land or locally? We have local people, internet people. Let's see. I'm looking for any information from anybody who's asked a question. Are you getting anything from your end, Walter?
Excuse me, no, I don't. No, in the chat, no.
Don't see any out there either. Oh, so Dan has a question. Go ahead, Dan.
Of course you do.
I just I just wanted to underscore this business with ACE2 and how specialized COVID is on ACE2. Is that COVID could have evolved 10 years ago in association with just one host and specialized on ACE2, but because ACE2 is 150 million years old and still exists in a lot of mammals, as soon as a virus specialized on ACE2 evolved in one host in one place, every host that had ACE2 instantly became a potentially compatible new host. So you don't have to believe that COVID is 150 million years old in order to understand that it can infect species representing a group that's 150 million years old.
Yeah, very
Thanks.
good point. Good point.
Thanks for making that point, Dan. I think I made the mistake of suggesting that COVID was 150 million years old. That's not
No,
the
you
idea.
didn't say that, but some people might want to infer from that, oh, well, because we know everything's got to be coevolution, then the virus has to be the same age as the host space.
Right.
That
Good point.
explains a lot of these
All right.
problems that we have.
It does. We have another hand up from BioInt. Go ahead, whoever BioInt is.
Hello, everyone.
Hello.
Good afternoon.
Very good. Go ahead.
Walter, thanks for your talk. I saw this term for the first time and I thought that it was very interesting. Parasite pollution. I'm not sure I understood.
Can you explain parasite pollution, Walter?
We are being exposed to parasite and pathogens, propagules all the time. Basically, that's what it is. We have viruses all around and larvae all around us, and they're trying. I start thinking about this when I went study in Idaho and I had swimmer's itch.
Yeah, I was just going to mention that. Swimmer's itch is one of the best examples of us trying to obtain something that we normally wouldn't hit. Yeah, especially in Nebraska, that's really common. So Dan, you had something else?
Idaho too.
Well, pathogen pollution also means that once a pathogen shows up in a certain place, it may cause an acute disease outbreak for a while, but even after the acute phase of the outbreak goes away, the pathogen never disappears. So once it's introduced, it stays there and accumulates.
It always hangs around. That's a good point also. All right, so let's see. Anything else from anyone? Go ahead and unmute and ask a question or make a comment or raise your hand. Okay, Kevin has one. Go ahead, Kevin.
Hi, Walter. Thank you so much for the talk. So yeah, I have a question. I know some.
This is Kevin Levano.
Yeah, hello. I'm Scott, PhD student. So yeah, my question is, I know there are some ancestral viruses with some sequences that are implemented in the, for example, arthropods that are currently like carrying those ancestral RNA viruses sequences. So, and there are like those elements from DNA viruses in all kinds of species, even mammals. Do you think that's part of these phylogenetic conservatism, or these conservative traits could be part of the host switching events in the nature?
I know if I got it, but it doesn't matter where it comes from. Any element of this capacity space of the species is what determines the ability of the species to explore new host. That's for sure. It doesn't matter if it's part of the genome of another virus of another species, and we know there's a lot of these going on these days. It just defines the capacity space of that species and the ability to do different things and survive challenges and take advantage of opportunities. Is that what you asked?
Yeah, well, it's just specifically related with the with these viral elements, like sequences that are like implemented in the host sequence, which are sometimes ancestral for viruses that they are even not here anymore.
So remnant pieces?
Yeah, well, like part of genes like controlling some expression of certain proteins.
Yeah, well, I
Very
can't.
little is known so far.
Not sure. I know we have a lot of these. I mean, there's a lot of exchanging of pieces of genome between different species and lineages in this planet. And I never heard of this. It's possible. I don't know if you know, Valeria is going like this. She probably knows it.
Yeah, we've been talking with her a little bit.
Yeah.
Thank you.
I can say I know.
Cool. So good to have some questions on the edges of stuff. So anything else? Anything else? Oh, Valeria, go ahead.
Yeah, and this is maybe a very silly question, but I think I'm thinking about this a lot, and I'm trying to find a way to formally measure historical compatibility. And from your example for fishes that you presented, it seems it may be measured as million of years or difference of million of years. I don't know. What do you suggest to formally measure this?
First time I think evolution happens in time. So maybe time is a good way of measuring, you know, what's the date of the last ancestor for two groups of hosts or two different species of hosts. I used Time Tree, you know, that software online that you can calculate the distance in terms of millions of years. So it's not a perfect measurement, measurement, of course, because evolution happens and you may lose compatibility. And if we look at the cladogram that I showed that of mammals and the ability of those species to maintain COVID and also shed COVID, it's different. So even though they're within all the mammals, there are some mammals that are apparently not capable of even keeping COVID going. They're not compatible with COVID. And so that means that they lost some level of compatibility.
It may be also like in the perspective of modeling, right? It may be also the interaction with opportunity being involved.
Absolutely.
In this
So we've been talking about this for a long time, right? I think there's no way we can actually understand this planet if we don't recognize that there are two elements, compatibility and opportunity, that have to be considered. Parasitologists like I in the past, we talk about host specificity. And sometimes we say that one species is highly host specific, but there's just one species of that genus in the entire area. and it's like a very distant genus from all the other species of fish in the area, the hosts in the area. So it looks very high host specific, but it was never exposed to a different species of that same host group, which means that the host specificity is ecological, right? It's not.
Yeah, I'm like taking notes because I'm trying to find this method. I mean, I didn't know that you are talking this today. You were talking about historical, and I have my notes here. It's 1 week. I'm trying to formalize this, and I think we will talk more about that for sure. Because the reality is that we need to simplify to model, but not too much. So I'm trying to find a better way to formalize.
That's Sabrina and Carla and everyone that in the lab knows that I've been thinking about this for a long time. I submitted like 3 or 4 different projects on this, and they were all rejected. But
of course.
But I think we do need to simplify, because the actors involved in an interaction are so distant from each one. I mean, we have all kinds of interaction. I mean, just look at fish with viruses, fish with bacteria, fish with nematodes, fish with monogenes, fish with whatever. The traits are not equivalent.
Yeah.
So we need to have a proxy for compatibility, even though it's not a perfect proxy. And that's why when I started reading about the theory of chaos, I thought it has a lot to do with whatever we're doing. Because first of all, evolution is not random, right? That means that there's a lot of things that are being inherited from ancestor and even those involved in compatibility. So we know there will be some erosion, there will be some changes, that's evolution, right? So some species within that group, we're not going to have the same compatibility. Sometimes you're going to lose compatibility, sometimes the compatibility will enhance. But at least you have a measure, at least you have a reference, you know, to say this is a potential host, or we are a potential host, for instance, for that specific virus. It is.
Yeah, I can see that. Yeah, thank you.
But
for-
at the same time, we have to remember that crazy things happen. I mean, just look at us as hosts of black yeast. And it's just because black yeast are thermophiles. So they can live everywhere, also in humans. So the biology is the science of exception, right?
Yeah, very good.
Thank
So
you.
very good points, Valeria, and thanks, Walter, for getting into it. I think that Valeria, when you come up with something, send us something so we'll have an idea of what your notes came up with. So, and Walter, thanks for being with us today. It was great, and it's good to see you. Haven't seen you for a while myself, so excellent. And please join us next week. We'll be here again, and basically, we say thanks so much, and we applaud your talk.
Thank you. I'm sorry
yes.
for the timing problem.
No, it was great, very good. So we'll talk to everybody again really soon. Be here next week, same spot. And
Good to see you all again.
yeah, let's keep in touch, okay?
Okay.
All
let's
right.
do it.
Bye-bye.
Bye.
Bye, everyone. And bye, Mom. My mom's here. So bye, Mom.
Bye.
Yeah. Ciao.